Welcome to video 4 and of course G08 on metagenomics. And here we're going to talk about metagenome assembly. So metagenomics assembly can take different approaches, and typically we're trying to take the appropriate reads to form context based on their relative similarity. But if we have large mixes of reads, that can be a complex process. So there'll be varied coverage of different genomes that are present, and then some may be very complete or very short. Um, the conserved regions that are shared across different genomes can be problematic, which is obvious, but a complex problem to resolve. And we also have to consider the diversity of the sample that present. We may have some serotypes or clades that are genetically very similar, but are all represented in the same sample. So typically we'll have short paired reads that we're looking at. So we can mainly use de Bruijn graphs for our assembly. If we're looking at long reads, we'd use the overlap layout consensus approach, which is shown here. That's looking at the pairwise overlap, but it's not maybe as computationally efficient as k-mer-based approaches, which take the reads, split them into k-mers of a given length, such as 51, typically odd, joins the k-mers together to create some sort of very big de Bruijn graph, which is iteratively proven based on information on the path structures and by remapping the reads to the contexts that are iteratively generated, we can create a more linearized set of contexts at the end. So if we have an in vitro assembly, we'd have typically one copy of each gene, or certainly even coverage if we did look at remap the reads back to its own context. If we look at co-assembly, the copy number of certain regions will vary, and it's not straightforward because some of the genes are shared. So co-assembly is different, which we'll do in part B, just taking the whole metagenome and assembling it all together. And individual assembly will try in part A by extracting the reads of Kraken2 first. An example of how our workflow, which takes in paired and fast queues, is Jovian. So it does QC, extracts human data because they're dealing with human data, assemble the reads into scaffolds and excludes the ones that are short. So typically ones that are less than 500 base pairs certainly are not worth exploring. It has to predict the orbs there, does taxonomic classification, aligns the reads to the scaffolds, and assesses the quality of these scaffolds. It also tries to look at look frequency variation that's present, and can therefore do create more advanced analyses at the end. This has been worked well on different viruses such as norovirus, enterovirus, rotavirus, hepatitis, HPV, Hep B, flaviviruses as well. So that's an example of approaches that are somewhat similar to what we're doing here. So here we can think of that we have a bunch of reads that we can. One option is co-assemble them context, and then we try to identify what species may be present using an approach. One of the typical approaches to look at the tetranucleotide frequencies. We're not doing that, but that's another one that's quite common. So with metagenomics assembly, we can think about our initial cyclone assembly of the short to great short reads. We may have different reference genomes that are present based on our mapping, and we're able to create context from that and take these mags to generate our metagenome assembled genomes. So part A of our lab is more like this because we're using Kraken2 to assist with this extraction of the short reads that are known. And part B will do de novo assembly of the whole metagenome to see what we get. There's different assemblers that can help us. We have Metafly that's very good for long reads. Petacomb, I haven't tried it yet, but can take short and long reads and is apparently a fully integrated pipeline. Then we have ones in the Spade family, which take short reads work fairly well for different data sets, whether it's metagenomic RNA viruses or metagenomic viral genomes, which we're going to look at as well. So we're going to focus on using metaviral spades for our practical because we're mainly focused on metagenomes from viruses. So Fly uses a slightly different structure to what we're used to. It makes a repeat graph from the long reads, and these are from imperfect k-mer matches. So it's not looking at exact k-mer matches because long reads are typically noisier. So it creates this kind of graph structure where we have different nodes and edges connected together, showing the linkage between the different graphs, and we can gradually extract information from these graphs to create some sort of consensus. And you can see that there's multiple joins here with different pieces of information in terms of the length and the coverage that is present. And that coverage information is useful in terms of that the coverage of the different contexts should be similar for if they belong to the same organism. So we have these sequences that are present and nodes that are present, and we can traverse. We obviously, like any assembly, we just traverse each node and graph once. So that concludes our topic here on metagenome assembly. Please look at the next part on metagenome classification.